Hello, everyone, and welcome to today's cryo-EM Current Practices webinar. You're joining the 3 national centers established by the NIH Common Fund Transformative High-Resolution Cryo-Electron Microscopy Program. My name is Christina Zimani, and I am a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. I am joined here today by my colleague at NCCAT, Ed Eng, as well as Lauren Vega and Nancy Meyer from the Pacific Northwest Cryo-EM Center. Michael Schmidt from the Stanford SLAC Cryo-EM Center, S2C2, is expected to join us as well. So if he pops in the room, he is with us from S2C2. PNCC has invited today's speaker, Tarjani Thaker, and Nancy will introduce her properly after my colleagues from each of the centers give a quick update on the cryo-EM resources we offer at no cost to the research community. And Tarjani is here from the Tomasiak Lab, and Thomas is also with us here up on the panel today. This webinar is a regular event that we host the last Thursday of every month, with the exception of November and December, as you all know. But starting again in January, we'll meet at the end of the month when S2C2 hosts Clarissa Dury from the OHI Lab at the University of Michigan on January 27th. And her talk is titled Structural Analysis of the H. pylori and L. pneumophilia type 4 secretion system core complexes via cryo-EM. So along with the exciting biology, all of the talks we host highlight the methods that researchers are using to obtain and interpret the cryo-EM data they can collect at the national centers. And we hope that these talks are a helpful resource, especially for groups just starting to use cryo-EM techniques in their lab for the first time. We are recording today's talk, and you can find that recording on our YouTube page or on the events page at cryoemcenters.org, where you can also find registration links for future talks in this series, and also general information about the larger NIH Common Fund cryo-EM program, including the multiple groups that are developing cryo-EM curricula and the newly established cryo-electron tomography centers. A couple of final logistics for today. Please use the Q&A feature to send in questions at any point during the talk, or you can upvote other questions that you see there. Lauren will be moderating Q&A at the end of the talk. And if you have questions directed to logistics or access at any of the centers, our panelists here will respond to them directly in that Q&A box. With that, I'm going to turn over to our center representatives to give quick updates, and I'm going to let Ed take it away first. Go ahead, Ed.
Okay, hi, I'm Ed here in New York at NCCAT. Everyone here at NCCAT wishes you a happy and healthy holiday season, and the best gift to give to your research program is the gift of cryo-EM. So investigators, for your consideration, please apply to NCCAT for either instrumentation access or embedded cross-training program or a screening program. For trainees, all these programs are available for on-site access. And this spring, we have our 1-week short course again. And so that'll be an immersive 1 week on single particle analysis, morning lectures, afternoon practicals. To find out more, follow us on Twitter at NCCAT.info or go to our website, nccat.nyspc.org, and I hope to see you in New York soon.
Thank you, Ed. And I'm gonna ask you to do double duty and just say a quick thing or two about S2C2.
Okay, so for my colleague, Mike Schmidt, who can't come right now, S2C2 is equipped with 3 Krios instruments, alpha, beta, and gamma. Alpha actually has a selectress in the Falcon 4. They also have other microscopes, including an Antarctica for training and specimen screening. Something that's upcoming in January is their user meeting, so I'm sure you'll hear about that soon.
Thank you, Ed. And now I will turn over to Nancy.
Hey, my name's Nancy Meyer. I'm a research associate and microscopist at Pacific Northwest Cryo-EM Center. PNCC is offering a single streamlined proposal type now for single particle and tomography projects, gives up to 480 hours a year over each project's 2-year duration. Proposals are accepted continuously and due by the 1st of every month. And each proposal gets delegated a scientific point of contact for project coordination. We currently have 5 microscopes, an Arctica K3, and 3 Krioses with Falcon 3 K3 detectors, 2 of these with BioQuantum K3s, and 1 Krios with a Falcon 4 BioContinuum K3. And 3 of these are now offering fringe-free imaging. For sample prep, we've got a Vitrobot and Leica GP2 plunge freezers and a Vitrojet that is right now still being put through its paces before its public debut. PNCC has been under COVID restrictions preventing on-site visitors, but we still offer one-on-one remote training and remote workshops on grid screening and data processing. And we're very excited and we're looking forward to in the new year allowing select visitors on site again and hosting in-person workshops. And you can visit our website for all of the details on all of the things and updates. Thanks. So I think for our main event today, I think I can go ahead and kick us off. Our speaker today is Dr. Tarjani Thaker, who is presenting recent work examining allosteric regulation in ABCC transporters. Together with her colleagues in the lab of Tom Tomasiak in the Department of Chemistry and Biochemistry at University of Arizona, their research focuses on a number of therapeutically relevant transporters. And specifically, they're using cryo-EM now as a tool to study conformational regulation, opening and closing, and drug binding in ABC and SLC family transporters. Tarjani received her Bachelor of Science at University of Delaware and Ph.D. in biochemistry at Vanderbilt University in 2013. And in the Iverson lab there, she worked on signaling mechanisms of GPCRs, as well as producing the first available structure of a eukaryotic acetate kinase. I didn't know that. And well, I don't mean to call her out as a crystallographer, but I think it's a compliment. And she has a great paper trail. So it's out. Her structural work continued as a postdoc in the Jura Lab at UCSF, and now as an adjunct professor at UA. She and Tom have really impressively ramped up their cryo-EM output, and we'll get a peek behind the scenes at some of their recent discoveries today. Tarjani is fantastic to work with, and I think they've really utilized our resources really well, and I think you'll like this.
Thanks, Nancy, for that introduction. I don't remember sending you a CV, so I guess you did some internet mining and found some pretty
out there
interesting information. Okay, I'm going to share my screen now. Let's see. Everyone can see this all right? Yeah. Okay, well, I am delighted to have this opportunity to kind of share where we're at in the Tomasiak Lab. As I was mentioning to Lauren and Nancy recently, we really would not be the lab that we are today without the access to the facilities and brain power at PNCC. So our group is in Arizona, in the city of Tucson, which if you're not familiar with where that is, it's a city just south of Phoenix. And I like to consider it the prettier city. So if you are thinking of Arizona as a dry, arid landscape, it's actually quite green here in Tucson, and we'll welcome anybody that wants to come visit and chat with us about science and collaborate and learn about some of our approaches. And so really in the Tomasiak Lab, the research focuses around understanding the complex biology of transporters in disease. And what that means is looking at alternate modes of regulation, whether it be from bound drugs or substrates, as well as chaperones and allosteric mechanisms by which they might alter their activity in specific disease states. And so the 2 classes that Nancy mentioned that we focus on are solute carrier family, SLCs, as well as the ATP binding cassette family, or ABCs. And both of these classes of transporters contribute greatly to cellular homeostasis. You can kind of see a summary of the main aspects of biological regulation that they contribute to. So we are interested in understanding how metals and metabolites contribute to metabolic rewiring in both cancer as well as certain types of infectious disease. And we also are interested in understanding how ABC transporters confer resistant mechanisms by mitigating oxidative stress pathways. And so both of these classes of proteins are linked to hundreds of diseases, and many therapeutics have been now developed targeting their function in not only humans, but in bacterial and fungal pathogenesis. So our focus really is on the ABC transporter family in the context of this talk. And so I'm going to introduce why we study ABC transporters and why specifically ABCC. And I'm going to follow that up by a little primer, which I think would be helpful for some of you here who are interested in learning about how to use PNCC resources to set up a functional lab in a somewhat of a cryo-EM desert. So that's where we are. And then lastly, I'm going to summarize some of the most recent findings, which will be soon hopefully published and available for you to all peruse at your leisure. So what are ABC transporters? So classically speaking, ABC transporters are a membrane protein family of regulatory proteins that are comprised of 2 transmembrane domains and 2 nucleotide binding domains. They can consist of either 1 or 2 polypeptide chains, and these come together to form this heteromeric association that acts as a scaffold for moving molecules from one side of the membrane to the other side of the membrane. And ABC transporters are unique in that their functions are linked to their ability to hydrolyze ATP in their nucleotide binding domains. Now, these topologies vary quite widely, and you can see that their subclasses are although largely structurally conserved, you can see that the connectivity and the diversity on this conserved scaffold varies quite greatly. And in eukaryotes, you have ABC transporters divided into 5 main classes, the A, B, C, D, and G family. And although this is a designation for eukaryotic ABC transporters, we can learn a lot about their function by studying orthologs in model systems like gram-negative bacteria, as well as fungi, which is the focus of our research. So the importance of these proteins comes from two primary modes by which they contribute to drug tolerance, which is a huge problem when we consider their function in drug resistance, right? So the first mode of drug resistance comes directly from the function of these transporters and their ability to efflux toxic molecules. And so one of the most famous and widely characterized ABC transporters, PGP, there's data for this transporter shown here on the left, which really illustrates how important it is in effluxing drugs in cancer and disease resistance. So these are size exclusion traces that were published back in 1976, and the peak with the asterisk above is showing the population that corresponds to PGP in drug-resistant cell lines of human CHO cells. And so at the bottom, you can see that this drug-resistant cell line CS53 has a huge overabundance of PGP. And this was eventually linked to its ability to recognize and efflux lipid-like and glycoprotein-like substrates. Now, BMRCD has been studied heavily as a model system for PGP. And you can see here this sort of direct mechanism of drug efflux, where looking at these giant unilamellar vesicles in the presence and absence of ATP, you can see uptake of the substrate in the presence of ATP, but loss of uptake in the absence of ATP or in the presence of ATP hydrolysis inhibition. And so this is a readily utilized readout for testing the function of ABC transporters, their ability to hydrolyze ATP and their ability to uptake lipid-like molecules in transport assays. Now, these are examples of direct drug transport, whereas in indirect drug transport, indirect mechanisms of drug tolerance stem from the ability of transporters like YCF1, which is one of our targets that I'm going to present to you today, in detoxifying cells from potentially damaging species. So looking here in this experiment, we're looking at different strains of yeast that have been either treated with toxic compound, cadmium, in wild type or knockout cell lines. And what you see is that knocking out YCF1 on the right in the presence of cadmium sulfate essentially blocks cell viability in this strain of yeast. But in the wild type strain, you can see that it confirms significant resistance to toxic cadmium levels in the cell. And so in this context, the mechanism by which this particular protein does this is through conjugating glutathione and converting oxidative species into non-reactive species and recruiting them in the vacuole. And so both of these proteins can inform on varied but important mechanisms of how ABC transporters contribute to drug tolerance as a means for better understanding their functions in human diseases that arise from drug resistance, such as cancer therapies that become ineffective, but also becoming more and more relevant in this current climate is really infectious disease, which is becoming an emerging public health crisis. And so how did these ABC transporters really evolve to develop unique functions and unique substrate specificities? Well, if you look at the sequence of just the nucleotide binding domain, you can see here sort of categorizing the main proteins that have been most well characterized in the ABC family. The nucleotide binding domain sequence is quite invariant. They all utilize very conserved elements to drive the recognition and hydrolysis of ATP in order to power the conformational changes that allow these proteins to pump molecules across the membrane. And so the diversity really comes from the transmembrane domain and variations on these scaffolds. So here we're looking at a topology of BMRCD and YCF1, which are proteins that study, compared to TMRAB, which is a ABC transporter that was one of Tom's main projects at UCSF, and now is probably one of the best structurally characterized ABC transporters. And both TMRAB and BMRCD are from bacteria. YCF1 is from Cerevisiae, Saccharomyces cerevisiae. And when you look at the topology here, you can identify this conserved structural scaffold where you have two halves of the transmembrane domain, you have nucleotide binding domains, and then you also have this conservation of asymmetry in its ability to catalyze ATP. So the first NBD binds ATP but cannot hydrolyze it, whereas the second nucleotide binding domain binds ATP and hydrolyzes it. So the power stroke really arises from the function of one out of the two NBDs. And in that sense, these are considered asymmetric heterodimeric ABC transporters. And when you compare the topology here, you can see that BMRCD and YCF1 have unique modifications to this conserved core. BMRCD has this extracellular insertion loop between TM1 and 2 that spans about 100 amino acids. And YCF1 has this really unique topology in which it has an accessory transmembrane domain, and it also has insertions, loop region insertions on the cytoplasmic side. And these have been characterized previously in systems like CFTR. And so the structural homology of YCF1 to CFTR is almost identical in that it has these accessory domains called TMD0, LASA motif, and R domain. And our goal here is to understand how these accessory domains in BMRCD and YCF1 contribute to their functional specificity and selectivity for substrate, and not just the conformational dynamics of how they move molecules across a membrane. And so when we consider that mechanism, alternating access is the model that's most widely accepted. And so in this model, you have a transporter that's in a resting state that's primed for substrate binding, primed for nucleotide binding. And occlusion of these sites drives the power stroke that allows the molecule to rearrange its structure and release substrate. So in this context, it looks like these transporters sample 3 states. But what we know is actually that they're a continuum of states. And prior to cryo-EM, which has really revolutionized our ability to study ABC transporters, which for the longest time really did evade structural studies because of the complexities of their dynamics as well as substrate specificity, scientists resorted to biophysical techniques like electron resonance spectroscopy. And so in this technique, what you can do is you can specifically label sites at important regions. So in this case, we can label, for example, BMRCD, 2 sites on the intracellular gate and 2 sites on the extracellular gate. And we can label them with labels that we can monitor as the protein interconverts between its various conformational states along its catalytic cycle as well. And so Hasan Musharrab has been studying this mechanism for as long as I can remember, and during my time at Vanderbilt. So Hasan is one of the leading deer spectroscopists in the country. He is at Vanderbilt, and he has spent his career really identifying what these conformational transitions are. And what we see here when we label this intracellular and extracellular gate, you see that there is a propensity for a primary state, but it's usually in the context of substates. So even in the wild type setting, when you look at the apo state, you can see this shift to an inward open, but you do sample these high energy states that have some slightly different inner atomic distances that correspond to either occluded or outward facing. The presence of substrate or inhibitor pushes these conformational landscapes to alternate states, but you still see a distribution of conformations. And so around the time that we were beginning this project, Hasan had published a really beautiful study on PGP, and we had been following his work on BMRCD. And what we found was that even in the context of mutations that limit its conformational flexibility and its catalytic cycle, you still sort of see this distribution of populations. And this is a little bit interesting because it conventionally speaking, sort of goes against some of the current accepted models for how nucleotide binding drives conformational changes for substrate release. And in the context of the catalytically dead mutant, we see that this distribution of populations is ever so slightly more discrete. And you can see in the presence of Hoechst, which is a model substrate for BMRCD, as well as ATP, you can drive this population distribution into what resembles a primarily outward facing state. And so a lot of these insights were exciting for us because they really helped inform on our approach to studying our main questions about ABC transporters and their regulation. And as we were pursuing these studies, which were really a tour de force for us being a small lab, we actually were delighted to see that one of Tom's former collaborators, Dr. Tempe, had successfully explored this conformational landscape using cryo-EM. And so this study really reinforced this, this idea that you have variations on this alternating axis mechanism, and that ABC transporters do not live in discrete conformational states, but they sample a continuum of states. And each of these likely contribute to both selectivity for substrate as well as substrate efflux. And so together, using the insights from the Tempe project, as well as Hasan Musharrab's recent studies, we sought out investigations into how conformations in ABC transporters like BMRCD dictate substrate selectivity, cooperativity, and transport. And so this is our primary team. I call these guys the co-founders of the Tomasiak Lab. And so our group primarily studies exporters, and we're focused on understanding selectivity and regulation. And our approach to this end has been to kind of integrate things that we've learned from past studies with new studies. So our approach here has been, as we developed our cryo-EM infrastructure, was to tackle something old. So Cynthia was the lead on developing TMRAB in the lab as a positive control for our pipeline. Something borrowed, we reached out to Hasan Musharrab, who was happy to collaborate with us on solving the structure of BMRCD, which is a protein he's been working on for over a decade. And then something new, which is Saccharomyces cerevisiae YCF1. And this is really Nitesh Khandelwal's brainchild and part of Tom's postdoctoral work under his R00 fellowship. And so I am happy to call myself out as a former crystallographer. Tom, myself, and Cynthia all have a background in biochemistry and crystallography, and Nitesh was our yeast biologist on board. And so together, we sort of put our heads together and were able to successfully determine not one, but two cryo-EM structures of BMRCD and YCF1 to better understand how drug selectivity is dictated by structure, as well as how regulation drives ABC transporter function. And so how did we get here? Well, I want to take a second to just quick acknowledge that coming from UCSF, we came from an institution with many, many resources, and we arrived at an institution with not exactly the same number of resources, but we were somewhat encouraged because all we really need in the phase one is a glow discharger and a way to evaluate our sample quality, which for those who have been at this for a while know that sample quality really ultimately dictates the success of your EM investigation. And we were a little less discouraged because our neighbor university, Arizona State University, just 90 miles to the north of us, actually filled some of the gaps that we were facing in infrastructure here at the U of A. And in the beginning, that 90-minute drive sounded somewhat reasonable considering Bay Area commuting traffic. An hour and 45 minutes doesn't really seem that bad, but as you start developing projects and as you start testing conditions for optimizing projects, this became kind of a limitation for us. And ultimately speaking, we didn't really have access at UCSF or University of Arizona or at Arizona State University just because of the number of groups that were trying to utilize these emerging techniques in cryo-EM. And so this is where we turn to PNCC. And really, without them, we really wouldn't have the insights that we have today. So how does PNCC enable us to perform cryo-EM investigations? Well, the proposal review process is absolutely phenomenal. It's very informative, and it's also quite quick. So although there's a formal process for applying, the review process, within 2 to 4 weeks, you have an answer. And as soon as your samples are ready, within 2 weeks after that, you're able to get into a queue. Now, not only is access important, but so is the expertise. So the 6 individuals shown here are the brain power that really helped us sort of validate our approach and also offered incredible insight into how to better improve our experiments. And so the process of data collection and data analysis is very collaborative at PNCC in a sense that things that we didn't know, we found out from them, and there was always an opportunity for an exchange of ideas and how to improve approaches, and even opportunities to help work with the developers to improve software. And last but not least is extremely high quality resources. So resources that are well maintained, that are operated by experts like Nancy and Theo and Drew, and also resources at the cutting edge of the technology. And so our lab actually opened in January of 2018, about 8 months before I got to Tucson, Arizona. And during this time, Tom really focused on reproducing anything. So protein expression, purification, optimization. This is the meat of our project was invested during this time, because without a good sample, you really don't have a rationale for moving a project forward in cryo-EM. And that's sort of our rule of thumb, is pay a lot of attention to sample quality, and then we can proceed to structure determination. And so in August, as soon as Tom kind of recognized that we were able to make proteins, after a few missteps, we ended up deciding to crowdsource funds within the University of Arizona to purchase our own plunge freezer. And so because ASU had a VitraBot, we decided to buy the Leica GP2, and this has been our real workhorse. It has been a beautiful instrument that has given us reproducible results for the last 3 years and has been working like a dream. And so this is the fifth member of our co-founding team, the Leica GP2. And so with that, we kind of eliminate one of these barriers to us being able to study structures using cryo-EM. And so once that GP2 arrived, we submitted our first proposal to PNCC, and within 2 weeks, we had an approval, and within 2 weeks after that, we were at PNCC, at least I was, with our first 12 grids. And it was really fortuitous, the timing of this, because this is where I sort of was able to take our samples and shadow some of the PNCC operators and the staff in how they use their Vitrobots and their GP2s. And so we worked out the nitty gritty of how to do plunge freezing on a instrument that frankly I had never used before on site, and was able to bring that expertise back to the University of Arizona, where now we couldn't imagine our lab without this instrument. So with insights into how to prepare samples, we sort of got to work, and within the month, by November, we were back at PNCC to screen all of our samples that we were able to get into cryo conditions. And we really approached this as a tour de force approach, a brute force approach, because we weren't really sure how access would pan out. Nancy likes to call us the beta users, which a badge I wear proudly. And I hope that our first few visits didn't really deter them from working with us, because although we screened 34 grids, I think I brought something close to 54 grids that sampled all 3 of our proteins of interest, our positive control, our something borrowed, or something new. And we left with 5 data sets, 1 for each day of the screening time that we had reserved on Arctica. And so during this time, we really got a feel for our grid freezing parameters or blotting times, our protein concentrations, and most importantly, because we're a membrane protein lab, detergents, and specifically how proteins behave in different detergents. So these data sets really kept us busy for the first half of 2019, where we took our data and we went back to the lab and we used it to build an infrastructure. Being the only real active cryo-EM lab at the University of Arizona, this was something we had to build from the ground up. We also purchased our first workstation and sort of utilized our ability to process quickly on our workstation, Panda, and validate some of the tools to build an infrastructure on our high performance computing cluster. And so what we found from our first 3D reconstructions during this time is a preference for detergent and ideal conditions for sample preparation. And so we went back in May to PNCC and we collected another 5 data sets, this time using image shift data collection, which allowed us to increase the throughput, which was something we wanted to test, because what we found in our initial round of reconstructions was a lot of conformational heterogeneity. And we weren't sure if that came from just sample instability or if that came from just the caveats of studying ABC transporters. And so with this data in hand, we realized quickly we needed more computational power. And like with any institution we've been at where we've relied on a cluster, the queue was something that we were facing a losing battle against. And so with Panda and Grizzly, we were able to process these initial data sets, get insights into our 3D models, and use these as a reference for how best to collect data on PNCC's Crioses, which between January to March, we were able to collect 4 data sets on 2 proteins in 2 different states using image shift data collection. And we left with about 23,000 movies. And this was done remotely. And so we were really lucky with the timing of this, because we all know what happened in March of 2020. Luckily for us, it was an excellent time to do nothing but process data. And lo and behold, our sort of 1.5-year commitment to improving our sample quality, really nailing down the conditions for sample preparation, building our infrastructure, and going into a Crios data collection with insights into what we were expecting to see, led us to publish these data sets. And so by spring of 2021, we had all of both of our papers on our structures in review, which are hopefully coming out within the next few weeks. And now here we are 2.5-3 years later, and we're on phase 2 of our projects. And we just bought our 3rd workstation, Yogi. So this is our sort of approach and pipeline. And I wanted to show this to give you context for how long it took us to go from sample to data. And PNCC was incredibly instrumental in keeping the timeline very, very short, but by being collaborative and being willing to work with us to try different collection strategies, we actually were able to solve structures from the first data sets we collected on the Crioses. So that goes to show you PNCC is an excellent place if you're trying to learn how to best collect data for a sample of interest. And so going back to our pipeline, we do focus heavily on optimizing sample preparation. So we're not so much concerned about sample quantity, but sample quality, considering we need very little amounts for structure determination. And so you can kind of see the benchmark that we have set here. We have this expectation for Gaussian peaks, as well as very monodispersed particles by negative stain. It's not always possible to get 3D reconstructions from these negative stain images, but we find that by confirming that at least the particles are homogeneous and well dispersed, that our issues of sample stability are likely not going to contribute to some of the downstream difficulties that typically arise with working with membrane proteins. So second, we're extremely detail oriented when it comes to our refinements, and our structures were really, we owe the success of our efforts towards developments in the computational side of cryo-EM. So one of the main problems that we faced was effects of the micelles, which we mitigated using side splitter and Relion, as well as using alignment-free and local refinements in system. So that allowed us to deal with structural and conformational heterogeneity. And lastly, but not least, we are very, very supportive of validating any of our models with cross-validation techniques, including biochemistry, so having a functional readout, bioinformatics, making sure the context of what we're studying matches evolution, and more recently, AlphaFold, which gives us an unbiased approach to testing how good our model building strategies are. And so with this, the first structure that we solved in the lab, this was my primary project, was the bacterial multidrug efflux transporter BMRCD. And so Hasan collaborated with us on this project, and together we sort of identified conditions for BMRCD in terms of mutants and detergents, and using Arctica data sets to identify which states were going to be most amenable for structure determination in order to kind of address these questions of substrate selectivity, as well as conformational cycles within a class of ABC transporter that bears a lot of homology to the C family in terms of its specificity for lipid-like substrates. And so from these three data sets, it became apparent to us that our wild type sample was missing something in its density. You can see that in the intracellular portion corresponding to the NBDs, we're lacking some density. We also see differences in the extracellular region corresponding to this loop insertion, but we were really excited to see that we could not only assign the transmembrane and the nucleotide domain, but we could assign this additional domain that we now refer to as the extracellular domain, even at the 200 kV level. And so we moved forward with data collection on a catalytically inactive mutant, which seemed to be more stabilized than the wild type. And we learned from our work with TMRAB and Dr. Tempe's work on TMRAB, showing the utility of mutations in stabilizing these dynamic proteins. But what we saw was that even in our QQ mutants, there were differences in the architectures that were somewhat unexpected. And there were also heterogeneities in the absence of substrate. So in the apostate, you can see we're still missing density in one of the NBDs, despite being a double mutant in the NBDs. And in the ATP molecule, we see a little bit better resolution in the transmembrane and the NBD regions, but if you look through the model, you can see that it's a little bit hollow in the transmembrane. And so these were kind of interesting to us because it gave us context for what we thought was perhaps a state, what drives stability, what is the high energy state, and what's the low energy state. And so our structure that gave us the best resolution structure was derived from a mutant of BMRCD that was saturated with Hooks as well as ATP. And so this first data set, we collected nearly 7,000 movies, and data processing on this required analysis of nearly 5 million particles that actually split into two classes, and two somewhat surprising classes. So if you remember the EPR data, we were expecting an outward open population to be stabilized in the presence of this mutant, and Hooks and ATP. But what we found was that this outward open population was actually way less resolved than at a second population that was this inward open state. And this inward open state, we were able to resolve the structure to 3.5 angstrom resolution using a pipeline entirely in Relion, which ended up being the one that worked the best. And that largely has to do with our ability to use side splitter to deal with micelle effects, using very large masks during refinement, and then applying particle polishing in the later steps to really get that last half angstrom improvement in resolution. And so with this approach, we were able to determine a 3.5 angstrom structure of BMRCD, in which we resolve both the intracellular nucleotide binding domains, the transmembrane domains, and now also this unique insertion in BMRCD, which is this extracellular domain loop. And so when we clip away one of the monomers in this chain, you can see that we also identify density in both nucleotide binding domains as well as the transmembrane domain. And this density we ascribe to 2 molecules of hooks and 2 molecules of ATP. And we were able to validate this by using masking around the ligand, no alignment-free classification, et cetera. And what we found was that this density was really, in fact, very robust. And if we take a closer look at it, the density was actually very asymmetric in nature. So we don't feel that it's an artifact of some solvent. It was primarily aligned along 1/2 of the molecule corresponding to the BMRC chain, which is also the catalytically inactive half. And so in order to better evaluate whether this was a real feature of our data set, we turned to Jose Feraldo Gomez's lab at NIH, and he's a fantastic membrane protein computational biologist. And what Jose did for us was simulate, based on our model, the energetic landscape of these 2 densities and whether 2 ligands of substrate could accommodate this pocket. And indeed they do in 2 independent simulations. And now what that raises the question of is, although the substrate is there, how do we model the orientation of this molecule? Because it is also, although somewhat symmetric, it's asymmetric in that it has 2 different polarities. And so using molecular docking simulations where we fix the pose of one molecule at one side and we allow the other side to sample freely, we were able to test 2 different orientations and found a preferential orientation at each site in alternating orientations. So essentially one molecule faced outward with its piperazine group pointing out and the other molecule faced inwards. And so what this arrangement led to was assignment of this binding pocket, primarily consisting of residues from the BMRC half of this structure. And what we found was that based on the molecular dynamics model and our model, we could identify a subset of residues that were overlapping in each. And so we tested these residues by mutating them and evaluated their effect on catalysis. So it's well known that BMRCD is substrate potentiated, which you can see here. And in the presence of hooks, you can see that the maximal rate is increased. And when we plot this as a concentration of hooks, we actually observe cooperativity. This cooperativity, when we mutate any of the given sites that are listed here, is essentially lost. They're lost to different degrees, but what we can tell is that there's no single mutation that preserves this cooperativity, which suggests that these 2 ligands might play an important role in dictating the conformational switch from an inactive resting state to outward to a high energy post-transport state. And so how is it possible that what EPR told us should have been an outward open molecule is resting in this inward open state? Well, we turned to investigations into this extracellular domain region, which our final model, you can kind of see the topology that we arrived at here. And what you can see is it fully occludes the extracellular gate of this particular transporter in a way that's very unique. So what we decided, and this was really an excellent suggestion from one of our reviewers, was to see whether this domain played any allosteric role in the catalytic cycle that powers the interconversion. And indeed, what we found was that when we label sites on the ECD as well as the transmembrane domain, that there's an ATP dependent interconversion of the conformation of the extracellular domain. And so if you specifically look at 126 to 55 and 126 to 44, you can see that there's a shift in the presence of substrate, whereas this is not seen when you look at the distances along the axis of the domain. And when we cut off this domain, we actually lose this substrate stimulation. So not only does it appear that this molecule has an insertion that drives substrate selectivity, but it also stabilizes it for catalysis, which is lost in the absence of this domain. While this is really fascinating, building this domain was a real beast, as we had absolutely no structural homology that I was able to identify based on sequence alone. And so our modeling was really driven by bioinformatics, EV couplings, Dolly searches of substructures within the domain. And while the density was adequately reasonable for assigning backbone structure, you can see that there's better resolution on one half versus the other. And this half, which corresponded to the beta sheet component of this ectodomain, was the most difficult to model. And so at the same time as we were refining the structure, AlphaFold was available. And so we compared the best model that I was able to derive to AlphaFold, and then use this model to refine against my cryo-EM maps. And you can see that there's a little bit of variation based on the map, but these are really at the termini and at the connection of the transmembrane domains. But when you compare the map to model fit, you can see that a drastic improvement in the correlation coefficient with the refined model using the AlphaFold structure compared to the one that was built somewhat ab initio. So this was a really nice, unbiased way for us to validate how to build our model to confirm the connectivity. And when we improved this final model and we performed a Dolly search, we now all of a sudden found a number of hits with structural homology, of which the most interesting happened to be penicillin binding protein, which matches almost nearly identically with the C-terminal region of this ectodomain. So although this domain has an unknown function, we've started to gain insight into its allosteric role in catalysis and substrate recognition, as well as transport, but also potentially insights into what kind of substrates this particular transporter is able to selectively bind. And so here I'm just showing a movie of our final structure that highlights the symmetry of the nucleotide binding domains bound to two molecules of ATP in the inactive and active site, along with the density of our two Hooks molecules within this transmembrane region. And you can see that they're in both asymmetrically positioned, but they also adopt an asymmetric orientation, and it's likely that it resembles some native substrate in this architecture. And so what we believe is the subject of future studies is how this enforces asymmetry and how it breaks symmetry in this class of proteins to drive the conversion from inward open to outward open. So our conclusions here are that we have determined a ternary complex of a model system of a ABC transporter that's important for direct drug resistance by transporting drug-like compounds. We have identified a structural basis for cooperative substrate binding in a model system efflux transporter. And we also now understand a little bit about allosteric regulation of ATPase activity by non-canonical domains. And so this is now an exciting template for us for future competitive inhibitors. So the second project, which I'll go through briefly because Nadesh will be presenting this at the Biophysical Society meeting, has to do with our work on understanding the allosteric regulation of a fungal ABC transporter, specifically by phosphoregulation. And so this is an avenue for diversifying ABC transporter functions that hasn't been necessarily well studied either mechanistically or structurally, because it happens to primarily occur on what people define as intrinsically disordered regions. And so Nadesh devised an expression system using an endogenous expression system, Cerevisiae, to express and purify native YCF1. And so what we found was that in this particular molecule, there was quite a bit of structural heterogeneity, which we were able to tease apart with careful classification. And so what we found was two primary classes that were somewhat marked by heterogeneities in the NBD regions, which for us was problematic because this is where we sort of we're mostly interested in determining the role of phosphorylation, and it's also the putative site for where this intrinsically disordered region of the transporter is thought to bind. And so through iterative refinement using side splitter and rely on alignment-free classification, and then local refinement in system, Nitesh was able to tease apart these two states into essentially 3 conformational states: inward narrow, inward facing transition, inward facing wide, to relatively good resolution. And so what was most surprising to us was that this protein adopts a topology very similar to the one predicted for CFTR, in that it has a TMD0. And within this architecture, we could also identify the arrangement of the regulatory domain insertions on this conserved scaffold, including the LASA motif, and the R domain, which is this putative intrinsically disordered domain. What dictates the difference between these two structures is not necessarily the architecture of these transmembrane regions, but the nucleotide binding domains in a wide versus narrow arrangement, where in the structure on the right, you have the 2 slightly more engaged than the 2 on the left, in a geometry that still nonetheless retains this inward open architecture. And so if we revisit this idea of regulation of ABC transporters by mechanisms outside of just substrate selectivity, we can actually find insights from sequence where there are putative phosphorylation sites on the intrinsically disordered region called the R domain that are conserved from yeast all the way up to humans in MRP1 and CFTR. And so what we found in our structure was not just resolution of these sites, but stability of this R domain in an orientation that gave us insights into how phosphorylation might promote intramolecular interactions. And so we found 3 primary phosphorylation sites, and we were confident of this because the density in this region was quite robust for phosphorylation. And we followed up this observation with a phosphatase treatment experiment where Nitesh treated YCF1 with and without phosphatase, lambda phosphatase, and found that indeed the signal disappears. So it appears that in our structure where we have resolution of this R domain, that phosphorylation at S914, T911, and at S908 are primarily driving the stability of this interface. And so when we take a closer look at this interface, not only are these electrostatic interactions between the phosphosites driving an interface, you also see organization of these networks at the transmembrane nucleotide binding domain region. So the LASO domain engages a region of the R domain. Then we also see regions of the intracellular loop 2, and a motif that we recently described in our JMB paper called the GRD motif, in engaging the R domain along the entire scaffold of the intracellular portion. And this putatively suggests to us a mechanism by which you could allosterically communicate changes in the nucleotide binding domain to the substrate binding pocket where glutathione and other presumed substrates bind. And so we were really surprised to see this R domain structure, which for the longest time has evaded structural characterizations for the thought that perhaps it was disordered. And so we again turned to AlphaFold as an unbiased way of validating our model building. And around the same time, there was a competing structure of YCF1 that was released. And what we found was that our structure of YCF1 in the phosphorylated state compared to AlphaFold not only conserved the register of the R domain, but it also had a conserved interface where the network of interactions that we observed between the R domain phosphosites as well as the hydrophobic pockets are present in both. And so this was really strong evidence for us that we have now elucidated how the R domain might help a ABCC family transporter like YCF1 interconvert from an inactive to activated state. And in this movie, what you can see is that if we just look at the morph between our 2 models, what's particularly surprising is that the geometry of the R domain remains constant. And so it really seems like the architecture of the R domain is focused on coordinating YCF1 for catalysis and allowing the geometry here to now adopt a conformation that's competent for ATP hydrolysis. And so from this structure, we were able to now state the structural basis for phosphoregulation through phosphorylation on disordered regions. This gave us insights into allosteric regulation of ATPase activity, again, in a related family of ABC transporter. And now with these insights and elucidation of novel hydrophobic pockets, these could also be used as putative allosteric sites for inhibiting YCF1 function and similar proteins in fungal pathogenesis. And so with that, I'd like to thank every single person involved. Our whole team in the Tomasiak Lab is phenomenal. We had an all hands on board approach to getting our cryo-EM infrastructure set up. Natesh and Cynthia have been side by side with me with grid freezing, with data curating, data processing. Tom has been on the back end, programming everything that we need, maintaining our workstations while also writing grants and papers. And we have a really amazing crop of grad students who are now part of our phase 2. This work, again, was possible because of our exciting collaborations through our collaborators at Vanderbilt and at the NIH. Obviously, our funding through PNCC and the NIH, which PNCC allowed us to kind of reallocate our resources in ways that helped us do the best possible science that we could without taking any shortcuts. And with that, I'll take any questions if you have them.
Awesome. Thanks, Tarjani. That was a great talk. We have 3 questions so far.
Okay.
So in the distribution of OF versus IF, do you think that the TMD1/2 could be in a dynamic motion, such as an NBD dimer and separates? They also wonder if you've seen any interesting movements from 3D VA or multi-body refinement. There's some other questions, but I'll let you follow. I'll let you finish with that.
Okay, so let me process this question. So I do think TMD1 and 2 is a little bit dynamic for a few reasons, because of the motion of the ECD that we observe in the deer data. And it's possible that that could be a sensor for dimer, NBD dimer versus NBD separate. And our structure likely informs on the NBD separate. It would be our dream to obtain the outward open structure to test this model. And as I was going through this talk, it occurred to me that maybe we won't ever see it without truncating the extracellular domain. And that might be a way for us to tell if that's the sensor or the allosteric link that couples inward open to outward open. I don't know if that answers your question, but that's kind of what I'm thinking. In terms of movements from 3D variability or multi-body refinement, we haven't done those yet. So this is on our list. Now that we've sort of caught up with our computational infrastructure, we had a lot of projects in the pipeline and not the most readily accessible computational resources. I am really interested in revisiting some of the data with the new developments in Reliant 4, as well as CryoSpark. So that's a great question.
They're also wondering if the concentration of intercellular magnesium ADP could affect the protein function.
Most certainly, yes. I mean, if it's, Tom, I might let you comment on this. What I would think is it really depends on the organism and the local concentration in that environment. In BMRCD, which is a gram-positive bacterium, I don't know exactly the what that looks like, but I would imagine that there's a balance of ADP and ATP, and that an excess of ADP might drive inhibition. So yeah, I think so.
Yeah, I can jump in too. So I mean, it's an excellent question, and these kind of issues about nucleotide concentrations, the ABC field fights a lot about. So you know, ATP concentration is pretty high in the cell, 5-10 millimolar or so. ADP concentration is high as well, so you always have it around. You know, so in terms of what its effect might be, you know, we don't quite know that, but we think the best way to find out is to do these experiments kind of the way Tarjani did, in the presence of a lot of ATP and magnesium chloride. So sorry, I don't have a better answer for you, but we think it's kind of always there, always saturated, right? At 5 millimolar, even though the binding's weak, it's going to have pretty high occupancy. So so these states where you look at it without maybe a little bit less relative or a little more fleeting.
Lauren, if you'll let me jump in for just a second, because we're
No
on the
problem.
hour. We only have a couple more questions. So if Tarjani doesn't mind sticking around for...
I'm happy to stick around and
Yeah. So
discussion.
we'll stick around for 5 minutes. If you have to head off to your next meeting, find the recording at cryo-EM-centers.org for the wrap up of these questions. And we wish you all very well in your cryo-EM endeavors. The National Centers are here to support your science. So let us know how we can help you, and come see us again at the January webinar. And I will let Lauren go right back to the questions.
I'm going to go to the next one. They're wanting to know what was important in to determine the detergents that you screened and ultimately selected.
Absolutely. So I have been thinking about how to concisely answer this, and I will say there's two things we focused on, not just the final detergent that we froze our samples in, but also the detergent we used to extract our proteins from membranes. And what we found was that LMNG with CHS was sufficiently adequate for extracting the majority of the samples in our lab for YCF1 and BMRCD. This is the case. We have new proteins in the lab where it's not the case. And that really contributed to stability. And the way we defined the stability was the peak Gaussian peak in size exclusion. Now for cryo-EM, we had in our Arctica data sets, we saw a lot of orientation bias. So we were just missing views. And so while we tested a number, well, not too many detergents, we tested 3 detergents. In every single case, digitonin was the detergent that eliminated these artifacts of orientation bias, and as well as improved a lot of our particle distribution on the grids.
Going back to your proteins, they're wondering if you've seen any lipid molecules, for example, in the space between the TMD1 first elbow that might be involved in function.
So Tom, cut me off if you don't want me to answer this, but in my maps, I do have evidence that there may be some lipid-like molecule. It's hard to say whether it's a detergent or if it's a lipid. But they are primarily at the interface of TMD1 and 2 and intracellular loops. So it's possible that there is a role for lipid in that mechanism that you're describing. Although it's hard for me to personally assign it because I'm very skeptical of assigning densities that are close to the micelle without higher resolution.
I would add that's also an excellent question. We do see it more in YCF1, which Min, I think that was your question. I just pulled up the structure you're talking about, which looks really, really interesting. And I noticed you have a couple of lipids in SIR2B. So YCF1 is similar to that transporter. And we do see lipids. We didn't build them in the model. The structure of YCF1 that was released before us from John Rubinstein and Wallach Canales, they also saw lipids in there. They assigned them to phosphatidylethynolamine, I think. And I think we saw them in the same place, but we were just a little hesitant to build them. So we didn't build them. We were a little cautious.
Awesome. So I have another question coming from the audience. And so one of them is, I wonder whether I'm going to slaughter this, Porsche molecules are exported simultaneously, and whether there's any functional data to address this. And then also, were there any extracellular domains, or what are the extracellular domains doing? And do these transporters import substrates?
So, so the extracellular domain of BMRCD is uncharacterized in terms of what its function is. And so this is the first structural characterization of that region. And as far as we can tell, there is no other structural homology within ABC transporters that I was able to find in the absence of related structures that look quite like that domain. But with our model, And we didn't have the chance to test this, but obviously this is very much a future direction. It would be really exciting to understand the relationship of that domain with the substrate binding and transport mechanism, in that the topology of that extracellular domain has electrostatic features that quite nicely match the substrate binding pocket. So it likely does contribute to some sort of substrate transport. I don't know if it's substrate import or substrate egress that it helps facilitate. I suspect that it probably does both, based on just where one would expect to find the types of substrates that the MRCD might encounter. But this is speculative and we obviously have to study it. Whether there's exported simultaneously, that's also a great question. So our cooperativity studies were really difficult because we really thought we could isolate maybe a mutation that preserved part of the cooperativity, but in fact, ablating any single site, we lost substrate stimulation entirely, suggesting that the two form not two independent sites, but a complete site. And so they're likely being transported somewhat simultaneously, but we'd have to come up with a biophysical technique to test that. Maybe Tom can add to that as well.
Yeah, no, it's that. Yeah, it's exactly it. I mean, we don't know the stoichiometry. We would love to do that. We will say there are more structures coming out, like PGP structure. There's BMRA structure now too, where we see multiple substrates binding kind of head to tail. So maybe more of a widespread thing in ABC transporters than we thought. But the stoichiometry question would be so cool to answer, but we don't have that info right now.
Yeah.
Awesome. Going back to some equipment questions.
Yes.
You know, obviously you guys did a great job covering how you built your resources, but can you comment on the capabilities and performance of your Leica GP2 relative to a VitraBot or other options for sample grid preparation?
Yeah, absolutely. So I will say I wasn't here yet when we bought the GP2. That was entirely Tom's decision. And I am a creature of habit. So if I had been in the lab, I would have probably advocated for the VitraBot. But the GP2 has been remarkable. So what I can say, having used both now, the GP2 maintains humidity really well. It's blotting because it's blotted what we've coined as like a face blot. So it blots head on to the grid. We also is an incredibly reproducible blotting approach. So one thing we found is that we actually had to add slightly more material to our grids than I think like classically people do. So instead of 2.5 to 3 microliters, we use 5 microliters. And so the way that the GP2 blots allows for all of that excess to be wicked away and I think partially reabsorbed. And what we found is that we can actually use way less material for the GP2. I'm surprised at the concentrations that we're using. And I don't know if it has to do with just being able to maintain humidity. The robotics of the thing are really good, or whether it's this blotting approach that works really well for us. But we're using in some cases less than like 3 mg per mil of protein down to 1 mg per mil per protein. And the only caveat of the GP2 is that the dewar for freezing is fixed. So what happens is if it gets icy, then you kind of just have to wait it out until it goes back. But the temperature controlled cryogen holder is excellent. The liquid nitrogen monitor is excellent. So we find that it's really, really high throughput and we can pretty much freeze. I think we've frozen like 40 grids in a day within a couple hours using the GP2.
Nice, that's awesome. So that's really all the questions that are coming in. We have some comments, which we'll share with you guys after the talk, but that's all we have. Christina?
I'm gonna stop my share.
Yeah.
No problem.
Absolutely. Yeah, we can wrap up now. So thanks for those of you who stuck on for those last questions. Thank you for that wonderful talk. Lots of detail, both about how to do the science and then just like absolutely gorgeous structions.
Thank you. I really appreciated this opportunity.
The ABC field is exploding faster with cryo-EM than I think anyone might have imagined. It's so exciting. So thank you very much. And please join us again next month.
Thank you very much. Happy holidays, everyone.
Yeah,
Thanks.
thank you. Thanks very much.